Thank you, Leslie, and thank you for that terrific introduction. It's a real honor to be here today. You know, the honors that are the most meaningful are the ones where they're chosen by your friends and colleagues. And so for that reason, it really is a meaningful honor for me, and I'm grateful for all of you to be here today. It's not often we get this many people together during a pandemic. Oh, wait, the pandemic's over. It's been declared by the governor. So, all right, so what I want to do today, whoops. So as Leslie said, my lab has worked on a lot of different things over the last few decades, particularly about the biology of RNA molecules. And I've had many terrific undergraduates, PhD students, and postdocs. And my apologies to all of them, some of them are here, whose work I won't even touch on, because I can't really cover too much in a short lecture. But I really want to talk about just two things. In the first part of the talk, yeah, see, this is what Leslie just says, just two things. No, wait, there's three. So, I'll talk about two things. One, first I want to talk about some of our work on how RNAs get degraded in our cells and how that led us to understand some new properties of a certain class of human diseases and how that led us to propose a possible therapy. And then I'm going to shift gears and I'm going to talk about neurodegeneration. This is more of a work in progress. This is a new area for me. As you'll see, it's a transition in our work. It'll be more of a work in progress, but I think it's important to talk about for two reasons. One, it's what myself and the members of my lab are thinking about right now, and so to be in real time, I want to talk about it. And the second thing is neurodegeneration is a big problem, and everybody in this room is being affected by it, either through themselves or family members or friends over the next decades. Just for bookkeeping, I have to mention I'm also a founder and consultant of a biotech company called Phase Medicine. What they do has nothing to do with this lecture, but I want to be completely transparent about my roles. All right, so to get started in talking about RNA degradation, I want to first remind all of us about how our genes work. And our genes really work in the following manner. We have information stored in our DNA where we have genes, and that when you want to turn a gene on, that gene is copied into what's called a messenger RNA. And this would be the same sequence of bases, the A's, G's, C's, and T's, and that encodes information to build a protein. And proteins really are the machines that carry out most of the work in our bodies. And one way to think about this, if you want an analogy, is you could think about the DNA is equivalent to a blueprint which is stored someplace in a library, and that when you want to use that blueprint, you copy it by Xeroxing it. Those of you who are old as me can remember what a Xerox actually is. But you copy it into a temporary copy, and then you send that to a factory to build the machine that blueprint encoded. And that's analogous where the mRNA is the temporary copy, and it's sent out to a factory. In this case, it's called a ribosome to build the protein. And I want to emphasize that there are really incredibly diverse numbers of proteins in our bodies. We have about 25,000 different protein encoding genes, and they make proteins which help us to digest our food. They make proteins which are what control and make our muscles work and allow me to be able to stand here. They make hormones which regulate our physiology. Insulin is one of those. It's what tells our cells to take glucose up out of the blood. And there are proteins which bind RNA. And that by binding to RNA, they allow the RNA to carry out its normal function and to be properly regulated. Now, because there's all these different proteins, our cells want to make them at different amounts and different times. And the way you can control that is by determining how much mRNA you make in the cell. So if you make a certain amount of mRNA, that is proportional to the protein that gets produced from that mRNA. But let's say I need more of that protein. Maybe I just ate a big meal and I really need a lot of insulin in my blood to tell my cells to take up the sugars from those meals. So I can turn those genes on and I can make more mRNAs. And that allows me to proportionally make more proteins. But the other way that cells do this is to not turn on the production of the mRNA, but to actually change the rate at which the RNA is degraded. And so if you make an RNA at a certain rate, but degrade it quickly, there won't be very much. But if you make it at the same rate, but degrade it slower, you'll have more RNA and you'll make more protein. Now, cells use all of these mechanisms to control our genes and the levels of mRNAs and how much proteins we make. But I've been particularly interested in how cells control this RNA degradation process. And so a couple decades ago, we really set out to answer this question, how do cells actually control the lifespan of mRNAs? And this just shows an experiment where we stop the production of mRNA at 0 time here. And you can see that this mRNA lasts for about an hour, whereas this mRNA is largely gone in a few minutes. Now, if you want to understand how something is controlled in our cells, you first have to understand what actually happens, right? Because you cannot understand the regulation until you understand the mechanism. And so we set out trying to address this question. And one of the great things about being a professor is that you work with incredibly talented young people, from undergrads to PhD students to postdocs to a permanent staff. And these people come into your lab, they provide the energy to do the experiments, the creativity to solve the problems. And these are some of the people who worked in my lab in the early years. And if you take all their work together, we can combine about 100 person years. It's 100 different individuals in the lab per year, into one cartoon. And this cartoon has a lot of things in it, but yeah, it's kind of sad, isn't it? But it's a really good cartoon. But what this cartoon shows are the molecular steps which lead to the degradation of our RNAs, and it shows the machines which carry out those steps. And because we now understood this process and we understood the machines that undertook these reactions, that allowed my lab and other labs around the world to begin to understand how different mRNAs last for different amounts of time and how cells control this under different physiological conditions. But that's not what I want to talk about today. Because when you come to an understanding of how some molecular event works in the cells, it often gives you new insight into things that you didn't know you were going to study. And so one of the things that we learned is that by understanding RNA decay, we came to understand how certain human diseases were caused. And this is based on a principle that cells like to clean up after themselves. And so to illustrate that, I want to imagine a healthy RNA. And this RNA, when it's made, goes about its function, and it does it reasonably well. And so the RNA persists, because at the same time this RNA could function, it could also be destroyed. But the destruction happens slower. So because this was fast, this never happened. But now let's imagine a defective RNA, an RNA which might have a mutation in it, which hinders its ability to do its function. This arrow slows down. Now this is the faster arrow, and the RNA gets destroyed. So there's no longer an RNA, and that's a way that the cell can remove these defective RNAs from it to keep the cell functioning. If you like analogies, the analogy here would be a broken down car on the freeway. This car is not functioning very well, and it's stopping up the traffic, and the solution that we use in human society is to get a tow truck to remove that car to allow traffic to restore its flow. All right, now, you don't always want to tow the car. Sometimes you have damage to your machine or your car, which you can fix. So all of us have probably gotten flat tires in our lives, and typically you can repair your flat tire in about 15 minutes, provided you know where your jack is, and be on the road again relatively quickly. But let's imagine you live in a city with predatory, unlimited tow trucks. And so as soon as you have a flat tire, within 30 seconds, a tow truck grabs your car, and not only grabs it, but it takes it to the car crusher. And so your car is gone, right? And so this leads to the destruction of potentially functional machines. It would not be a good thing. This is exactly what happens in a class of human diseases that I'm going to tell you about. And so these are what we call RNA hyperdegradation diseases. These are cases where something happens to the RNA so that it could function, but it never gets a chance because the predatory tow truck grabbed it and took it away. And so in a normal situation, let's imagine an RNA that binds its protein, and that binding creates a normal function, and everything's okay. But now, maybe there's a mutation in the RNA, maybe a mutation in the RNA binding protein, so that these come together slowly. You get little of this normal complex formed, and this competing RNA degradation pathway destroys the RNA, and there's no function. And if that RNA was important for the cell, then you have a problem, and that can lead to a disease. And one of the examples I want to talk about today is a disease called dyskeratosis congenita, which occurs when the telomerase RNA undergoes this kind of process and gets prematurely destroyed. Now, the telomerase RNA is a really interesting RNA. This is an RNA that forms a protein complex, shown here, and in a healthy individual, this forms and everything's okay. Now, this RNA is different from the messenger RNAs I talked about earlier. And this illustrates that in some cases, RNA can be part of cellular machines. I told you that proteins do most of the work in our cells, but there are special RNA molecules that also contribute to how molecular machines work in our bodies. And this discovery that such RNA machines worked in our cells was really made here by Tom Cech at CU Boulder, and this is what led to his Nobel Prize. So we're building on good roots here. All right, so what happens in people who have dyskeratosis congenita is you get a mutation either in the RNA or in one of these proteins. You can't build the normal complex, and the RNA gets destroyed. Now that's a problem because what the telomerase RNA and protein complex does is it extends the ends of our chromosomes. We all have DNA in our cells, and every time our cells divide, the telomerase RNA protein adds a little bit of sequence to the end of our chromosomes. And if you don't do that, every time your cells divide, they get shorter and shorter and shorter and shorter until they're too short, and then the cell just goes through a catastrophe. So you need the telomerase enzyme to keep your dividing cells functional. And this is most important in cells that divide frequently. Our skin cells divide at such a rate that every 27 days you completely repopulate your skin. So people who have problems in this telomerase enzyme have all kinds of skin problems. The place that's more important is your bone marrow. Your bone marrow is a rapidly dividing set of cells which provides all of your your blood cells, both white and red, over the course of your life. And patients, dyskeratosis congenita patients, usually end up with bone marrow failure in their early 20s because their chromosomes have gotten too short, they can no longer function, and now they can no longer make red and white blood cells. And there's really no effective treatments for this disease at this time. All right, so a couple of years ago, this student, Sid Shukla, was a CU biochemistry PhD student, said, well, maybe we can understand how this telomerase RNA gets degraded. And if we could understand how it gets degraded, if we could block that, maybe we could treat this disease. And this was a work helped by Jens Schmidt, who was a postdoc in Tom Cech's lab, who's right across the hall from my group. All right, so what Sid learned is that in normal people, the telomerase RNA binds its proteins and everything's okay, although there is this other event that can occur. But in people who have dyskeratosis congenita, either due to mutation in the RNA binding protein or sometimes in the RNA, you can't make this complex effectively. So this other machine called PAPD5 comes in and adds a bunch of A's to the end of the RNA. These A's are a tag to say degrade this RNA. You can think of this just like a logger going through the forest and putting ribbons on trees that the tree cutter should go and destroy when he comes through. So this tag that gets added recruits an enzyme, the RNA gets destroyed, and then this individual ends up with bone marrow failure and lots of other problems. So we understood what happened, but then we got really lucky. because it turned out that several biotech companies had already developed drugs which inhibit this enzyme, this machine. And they had done this because this machine, this PAPD5, is important for hepatitis B infection. And so people cared about trying to develop drugs to treat hepatitis B. So Sid said, wow, look, we have these drugs. Can we use these drugs to effectively treat dyskeratosis congenita? And so he showed that would rescue the telomerase RNA in cells from these patients. And then he asked if he could restore blood cell formation. And he did this by working with a guy named Luis Batista at Washington University in St. Louis. And what he did was take advantage of what are called human embryo stem cells. And I want to back up and remind everybody, these are cells which essentially have all the properties of cells in very early human embryos. And so they can, under the right conditions, be coaxed to develop into blood cells, skin cells, muscle cells, whatever you want, because that would happen in a normal human embryo. And what Luis can do is take those embryo stem cells and in the lab, turn them into blood cells. And that works quite effectively, and he's really good at it. But if he takes a cell which has one of these mutations causing dyskeratosis congenita, it doesn't work. You can't make blood cells. And of course, the key experiment then is if we add this drug which blocks the telomerase RNA degradation, we can now completely restore the production of both red and white blood cells. So this is really terrific. It didn't have to be this easy. Most of the time you have ideas like this and you go and you test them and they fail miserably. So this is really terrific. Will this really lead to an effective therapy for this particular disease? I'm optimistic for two reasons. One, when we add this drug, not much else happens to the cell. It doesn't change many other RNAs. It doesn't really change much of anything. And I can talk about why that is if anybody's interested. The second is another lab recently showed that you could actually use the same kind of approach to rescue blood cell production in a mouse model of this disease. And so that suggests it can actually work in an organism, and that's one step away from trying this in people. And so I think the next step here is to actually try this in people, and I know of at least 2 biotech companies which are trying to pursue that approach. All right, so I just want to kind of wrap up here and make a bigger point. Really we started out a couple of decades ago just trying to understand what happened to these RNAs? Where did they go? This is really fundamental basic research. We had no idea that we were ever going to have an impact on the clinic. But yet by understanding what happened, the mechanism, it allowed us to have insights into what might be going on in some human diseases. And then that has now even led to the idea of a possible therapeutic intervention. And I think this is I just want to emphasize how important it is to do this groundwork, this basic research, because without that, we never would have got to this point. And so really, when money gets tight in research, often we try to stop funding basic research, but it's really important that we keep doing that. My lab continues to do this. We work on some other hyperdegradation diseases that we can actually treat the same by similar strategies. And we actually have now started working on what we call hypodegradation diseases, where the RNA lives too long, and if it lives too long, it can gunk up the problems and make it and cause issues. So this is where we all take a breath before I make a transition, because I'm tired of talking, so you must be tired of listening. All right, so then I had a midlife crisis, but in terms of science, not in terms of my life. This happens in science. If you're making progress at some point, you have to find something new to study, right? And if you're not making progress, maybe you should find something new to study anyway. And I was getting older, and I started to have friends and family who were affected by neurodegenerative diseases. And as I started learning about them, I realized this was an incredibly major societal problem. It's very poorly understood in terms of the biology, so there's a place for someone like me to make a contribution to understanding what's actually going on. And there's some connection to RNA biology, which was poorly understood. And so I decided to shift my research into studying neurodegenerative disease. And I just want to emphasize, this is really a big problem. And I want to make a distinction between dementia, which is really a set of symptoms, and such as memory loss, difficulty completing tasks, disorientation, and neurodegenerative diseases, which are often the cause, but not always, of dementia. And this is the 6th leading cause of death in the US, and that's actually just counting people who die from Alzheimer's disease, not the other half of people who die from dementia. It's very expensive because people who have these diseases can live for quite a while. And it's also incredibly emotionally difficult for the friends and families and caregivers of people with neurodegenerative diseases. It's getting worse, right? We as a society are all getting older. This is the age distribution of our country in 1980, and you can see there was only 4% of the people over 75 years old. And in 2050, roughly 13% of us are going to be over 75. And I hope to be one of those. The rates at which you get neurodegenerative diseases go up with age. That's the #1 risk factor. And so more old people, we're going to have more neurodegenerative disease, and we have no way of actually treating these diseases right now. Now, I want to step back and talk about neurodegenerative disease a little bit, but I want to emphasize that these diseases are incredibly complex diseases. They're affected by things like neuroinflammation in your immune system. There's an interplay between different types of cells in your brain and how that affects this. Your cardiovascular health can have a big impact on some of these diseases. And there's genetic environmental inputs and on and on and on. I can't talk about all those things today. So what I want to do is just talk about what I think is the crux of the issue in these diseases and how we're trying to address them in my lab. And please realize this is going to be simplified, and I'm happy to answer questions about it in the follow-up. The first thing I want to emphasize is that neurodegenerative diseases arise because neurons die. And if you look at a brain from someone who had a neurodegenerative disease, it's smaller and regions of the brain have shrunk, and that's because your neurons have died. Our brains do not reproduce our neurons. The neurons you get when you're born are the neurons you have today. And if you lose too many because they die at some rate, then your brain function is compromised, and if you lose enough, you can actually end up with death. By and large, neurons die in these diseases due to the buildup of what are called aberrant protein aggregates. These are sections of a brain from various different neurodegenerative diseases, and they're stained to show these brown clumps, which is a staining approach clinicians often use. And these are showing not normal, aberrant or not normal protein aggregates that form in all these diseases. This is a common theme of all these diseases. There are different proteins. In some diseases, it's this protein called FUS. In others, like Parkinson's, it's a protein called alpha-synuclein. And in Alzheimer's, as I'm going to talk about, it's a protein called tau. So different proteins, different regions of the brain, but the common theme is this aggregation of proteins to cause a dysfunction of the neurons. So because as these proteins clump up and form together, they're often referred to as what are called protein folding diseases. And I want to step back and remind people, proteins, when they're made, are strings of amino acids, and they fold up into a structure which what allows them to have their proper function. And if you like, you can think of this as a chair which has been assembled correctly so that you could actually sit in it. But these proteins that form these aberrant toxic clumps are really misfolded. And I was lucky enough to be at Leslie's house when Mike was building a chair from IKEA the other day. And you can see he put together a misfolded chair. So this is what these things look like, all right? All right. So they're misfolded proteins, but that wouldn't be so bad, frankly. If you had a few misfolded proteins here or there, our cells have ways of dealing with that. There are two things that make this really a problem. Here I go, Leslie. The first is that the misfolded proteins that cause disease spread. So you don't just get one misfolded protein, you get a whole bunch of misfolded proteins. This is by what's called a prion mechanism. And a prion mechanism occurs in the following manner. You have a whole bunch of normally folded proteins. This is what they normally look like. But one of them, through some event, forms an aberrant structure. That aberrant structure can interact with other normal proteins, convert them to the aberrant structure, grab another normal protein, convert it to the aberrant structure, and so on and so forth until you end up with this huge pile of aberrant proteins that fills up the whole cell, and the cell eventually dies. If that's confusing, there's an analogy to this in our political culture where the entry of an aberrant politician can trigger other politicians to enter that toxic state, and that can propagate with some variation until eventually democracy perishes. All right. Okay, so that's one problem. The problem being that these proteins, when they're aberrant, they spread. So it's not just one event, it spreads. And not only does it spreads between neurons. So imagine a neuron that has some of these aberrant protein folds in it. That neuron's eventually going to die. When it dies, it releases what are called seeds, these little parts that can start the whole process over again. They get taken up by a nearby neuron. That neuron then builds a whole bunch of these aberrant structures. It dies and keeps the process going. And so this prion propagation is what leads to the progressive but slow death of more and more and more neurons over years, and eventually leads to the decline in function and death that occurs in these diseases. All of these proteins spread by prion-like mechanisms. That's rather amazing to me, right? And I would be willing to wager that all these neurodegenerative diseases that spread and are progressive are going to require this type of spreading mechanism. We know that you can have damage to your brain that does not spread. You can have a stroke, which kills part of your brain. But generally, that's it. It doesn't spread throughout the rest of your brain. It just, you have an acute damage, and then you figure out how to go forward. And so this prion-like spreading is really the crux of the issue, because it's what drives the disease and allows it to propagate and kill more and more and more neurons. I want to focus on this one on the right here, tau. Tau is a protein that aggregates in Alzheimer's disease, but it's also central to over 25 different neurodegenerative diseases. In fact, 80% of the dementia cases in our country are caused by aggregation of tau. So it's disproportionate, there I showed you 4, but this is the big dog in terms of causing disease. This occurs in Alzheimer's disease. It occurs in chronic traumatic encephalopathy. This is what's called CTE. This is the disease that football players get when they repeatedly get banged in the head, and it eventually triggers tau to start this prion propagation mechanism, and then it leads to death of their neurons and disease. This also happens in some infections. If you get infected with measles, 1 out of 500 to 1,000 people end up getting, they recover from measles, but they end up getting one of these tau propagation diseases, and they die 10 years later. So if you know people who think their kids don't need to be vaccinated for measles because they're going to be okay, measles isn't so bad, there's a 1 in 500 chance that that child is going to die 10 years later after they get measles. And I don't think people appreciate that. I wouldn't take that chance with my kid. All right, so we've become very interested in tau. Oh, and I want to mention, many of you in the room, I'm talking about tau as the cause of Alzheimer's disease, but many of you, of course, have heard about A beta, and A beta and the plaques that it forms as a causative event in Alzheimer's disease. And that is true. This is another protein that forms protein aggregates. They're outside the cell, and the best understanding of this right now, from not just me, but from the field, is that really what happens when you form these A beta plaques is they actually trigger tau to start this aberrant propagation and prion-like mechanism, and then it's the spread of the tau aggregation which leads to neuronal death. So A beta is involved, but it's upstream of tau, and tau is really the crux of the disease. All right, so we've gotten interested then in how this works, and I think some of the really key questions then, what are these tau aggregates? How do they form? What controls that process? Can you manipulate that so that you could actually prevent tau aggregation? or their toxicity to eventually lead to some kind of clinical intervention. So why me, right? There's a connection to RNA here. If you look at these 4 proteins, 3 of them are really good RNA binding proteins. I've always wondered about this. For 20 years, I've wondered, why do all these proteins that cause disease bind RNA? And then as I'll show you, there's some connection to RNA biology that people just don't understand. So that got me interested in this, and I felt, well, it's involved with RNA. Maybe we can do something. And a few years ago, an MD PhD student joined my lab, Evan Lester. Evan was fearless, to say the least. And Evan said, well, maybe I should study tau and see if tau aggregates really are actually involved with RNA. And so, his logic was, many of these proteins that aggregate are in fact RNA binding proteins. And stuff I haven't told you today from work in my lab is that RNA is also really good at forming aggregates. If you have RNAs which are not doing their normal thing, they can actually interact through base pairs to form what we would call an RNA aggregate in cells that might have some problems. So Evan's PhD was to see if there are RNAs in tau aggregates and could they explain some of the toxicity. Now, how do we study neurodegeneration? Despite the fact that we'd like to be able to look into the brains of some of our colleagues, we actually can't do that. So what we do is use simple model systems. So we use mice, and these are mice which a normal mouse with age, their brain stays pretty healthy. But if you genetically engineer a mouse to make these tau aggregates by introducing such mutations which cause that, then over time this mouse develops these tau aggregates and gets problems very similar to what humans do. We can also take those mice, and if we take extracts from their brains, this one's not going to have anything in it, but this one's going to have these prion-like seeds, right? Because there are tau is propagating in there. And if we transfer that brain extract into cells in the lab, then these cells will start making new tau aggregates. And this allows us to begin to study how this propagation occurs in cells simply and efficiently in the lab. And that's how we do a lot of our experiments now. Now, finally, we have to go and look in people because in the end, a mouse is not a human and a cell in the lab is not a brain. And so if we learn something in our model systems, we have to go and see if it's true in human patients. And here we've been very lucky to work with Stan Prusner's lab at UCSF and also Nadine Bakker at the BioFrontiers Neurological Institute, who have been terrific at helping us with either mouse models of disease or providing us access to brains from normal and diseased individuals after they passed away. All right. So Evan, I just want to say a few things about what Evan learned. Evan learned that in fact tau aggregates do contain RNA. This is a tau aggregate he purified out of, I think this one's out of a mouse brain. You can see if you look at make tau turn green and RNA turn red, it's full of RNA. For the aficionados in the audience, it's actually mostly full of snRNAs and SNO RNAs. It also contains a bunch of RNA binding proteins. And we didn't anticipate this, but this one here called SRM2, you can see normally here the red color is in the blue region. This is the nucleus of the cell. This is where the DNA is. And this tau aggregate here is outside of the nucleus. And you can see that the red protein left the nucleus to end up in the same place that tau was. This was a surprise. Is this really meaningful? So the first thing we did is went and looked in human brains. And this is an experiment done by Felicia Oy in Stan Prusner's lab, where we took a brain from a patient who had cortical basal degeneration. This is a disease where they develop tauopathies in the central region of the brain. And if you stain for tau, you can see the normal cell doesn't have any tau aggregates, but this patient has these tau aggregates throughout that region of their brain. If you look at the red protein, it's actually in the nucleus where it's supposed to be in the normal person, but in the patient, it's actually now moved out to be in those tau fibers. So the same things happens in patients. And this is true not only in this particular type of disease, it's also true in other tauopathies such as Alzheimer's disease. All right. So why do we care? And we care really for two reasons. One is we think this might be telling us something about the mechanisms by which tau aggregates actually kill cells. Normally this protein's in the nucleus and it plays a role in making new mRNAs. But when it's gone from the nucleus and out here in the cytoplasm with tau, that leads to alterations in how cells make new RNAs. And those alterations are seen when people study post-mortem study samples from the brains of people who died of these types of diseases. So there's a correlation with what we would expect from this with what actually occurs in diseased patients. So that's one thing, and we're trying to continue to see if that's really true. The other reason we care is this may tell us something about how tau aggregates form and grow. And this is, I think, to me, one of the key questions is, how do these things actually grow? How do cells make these aberrant structures? Because if you could understand that, maybe you could change it. And I'm going to suggest a possibility that maybe tau aggregates grow off some kind of assembly which contains this SRM2 protein. Now, how do we understand where tau aggregates form and how they grow in cells? You know, what we have is a picture before where there's no tau aggregate, and we have a picture after the fact where there's a tau aggregate in the cell and it's got some of that red protein in it. But how did it get there? This is very analogous to watching a soccer game where you might have just a picture of a midfielder with the ball and then a later picture with a goal, and you don't really know what happened between those. But if you can make a movie, what you can see, of course, is that Pratt does a long pass here up to McCann. McCann goes around Decker, goes around, shoots goal. So, okay, so you can see what actually happens. All right, so can we do that for tau? So we can, and I wanted to show this because I think there's a kind of a revolution going on in biology where we can actually watch things happen at cells because of the ability to both add colors to proteins. So we can make tau turn green, and I'll show you a movie where we've done that. And we can make this red protein, SRM2, turn red, right? So we can actually see them. And then there's incredible new microscopes that can take really sensitive movies and can image cells multiple times per second. And in fact, one of the things BioFrontiers does is run an imaging facility where we keep, kind of up-to-date new microscopes available so that people all over campus can come and do these kinds of innovative experiments. All right, so let's make a movie. So what you're going to see here, I'm just going to play this. Watch this cell in the middle. So first thing you'll see is a little red ball form. Okay, and then on that, wow, look, tau grew. Tau grew on that little red ball that formed first. This cell eventually gets really ugly and dies. All right, so what does that really mean? What's really going on there? And I'm going to show you that movie again, but first I want to show you the cartoon of what we actually think is happening. What we think is happening is that there's an assembly that forms in the cytoplasm. It forms transiently, and it contains some RNAs, and it also contains that protein SRM2. Tau somehow likes to interact with that. It likes to go to the surface of it for some reason. We don't know why. But then once it goes there, that surface is really good for some biochemical reason at helping that tau aggregate to grow. Right? So now that's what we think is going on. Let's watch the movie again, and you can kind of see that the first thing that happens, we'll start here, then we'll go here, and then we'll go here, and we'll just keep growing and eventually lead to problems. All right. So why do we care about this? I really care about this because I think the whole crux of these diseases is how do these aggregates propagate and form more aggregates and get into new cells and form more ones and spread throughout the brain. If you just lose a neuron here or there, you're actually okay. But if it spreads and you continue to kill more and more neurons, that's a problem. And so we're very interested in the lab at trying to understand what is it about these structures which causes tau to go there? How does it actually promote tau aggregation? What's going on there? If we understand it, can we change that so that we could alter tau aggregation and potentially influence how the toxicity would occur? And then we're also interested in more experiments I don't have time to talk about today about the role of RNA in these aggregates and what they might be doing. All right, so I just want to wrap up now in the last couple of minutes. Now I've kind of tried to tell you two things about all the different things we do. One about RNA degradation and how it plays a role in kind of prematurely getting in the way and degrading stuff that shouldn't be degraded and causing some diseases. And then I've told you a little bit about some of our newer work on RNA biology and neurodegenerative disease. This is something that will be a major focus of my lab over the next several years, and I hope that we can make some meaningful contributions there. I want to end by really acknowledging my entire lab, many of whom are here today. Receiving an honor like giving this lecture or being here is not really about me, it's about the culture and the individuals in the lab and the community of a lab, which allows people to synergize, to work hard, to be creative, and to solve problems. And I have a terrific lab and have always had a terrific lab that's really contributed to the success of our experiments. So thank you all. So I can't really give this talk without talking about a woman named Denise Molard. Bear with me for a second. Denise joined my lab in 1990. First person I ever hired. Was in the lab when I had brown hair and no gut. We raised kids together. Her daughter's a few years older than our sons. And she moved to Colorado with me a decade ago. Denise passed away tragically in 2020 in the fall from an unknown cardiac abnormality. It's very sad. But she was really the heart and soul of my lab. I thought I could do this. But she really created a community, and she saved so many PhD students because your dissertation can be really hard. She was the rock that was there to help them through the hard parts. She remembered everybody's birthday, made everybody's cakes for their significant events and their PhDs, and whenever somebody had a child, knitted incredible quilts or bags to give them to them. So she was actually a terrific member. Those of you in the lab who know Denise, I think, will attribute to this. She's taught me how to be a better person, and I can't talk about the work in my lab without giving her a big debt of gratitude and acknowledging her important contributions. Okay, so now I'll take questions. Right? Thank you.
Roy, for that really wonderful talk and a really wonderful tribute at the end there. So at this point we have questions. We have microphones for those of you who have questions. I know there are a lot out there.
Hi, thank you for the wonderful talk.
Thanks.
Is it known whether the prion process has a useful function that's like sort of fundamental to the way the brain works and these things are sort of hitching a ride on it? Or is it something that's some weird thing that never showed up before these things were discovered to be using it?
Yeah. So I would answer that question in two ways. Certainly it's true, this ability of a protein to form a structure and then convert other related proteins to form that same structure is used in biology to do important biological regulatory events. So this kind of molecular mechanism is part of normal function in biology, okay? Having said that, it's also something that lots of proteins can do. And there's no evidence that the proteins which do this and cause disease do it as part of their normal function. The proteins that do it as part of their normal function are other things. So I think and I suppose we don't really know the answer yet because, to be frank, we don't know what the tau protein does. Despite the fact people have studied it for 20 or 30 years, nobody really knows what its function in our brains is. So my guess is that this is not part of their normal function, but I would say stay tuned. So,
Yeah, thanks again for this very nice talk. If I can go back to your first story with telomerase, have you any idea of what triggers the proclivity for degradation of that? You've blocked it at the end point, but what's going on to target those molecules for rapid demise?
right. So I think it's really just, I'm going to go back to that cartoon because it's easier for me to show it than to say it. So what we think is going on is that this reaction can occur all the time, but if the protein's there, this occurs faster. And you can think of the proteins essentially as binding and getting in the way and preventing this tagging enzyme from tagging the end of the RNA. But if you are missing those proteins or have mutations here, then this thing is just there all the time, and it now adds the tag, and the RNA gets degraded. So it's really, in a way, it's about a kinetic competition, and it's just about the rate at which this happens versus the slower rate at which that normally happens. Does that answer your question?
So the proteins are really there to control the degradation, not for the function, the basic function of the telomerase.
That's a really interesting point. So we didn't know that going in, but I think you're absolutely right. It could have been that if you didn't have this protein, even if you made the RNA stable, it wouldn't work because you needed that protein. But you're absolutely right. The fact that we can not have this protein, but we can rescue function, says you don't really need that protein for function. You just need it to keep the RNA around. And so that's actually a really good point. Yeah.
Thanks, Roy. More than two good things. So I'm interested in the prion propagation. What is known about the cell-to-cell spread? Why doesn't that happen?
Yeah. So what Kristen is asking is, how do these molecules what's this what's known about how this occurs, right? And so there's a number of people who've been working on this. There's a receptor, which Ken Kozik's lab has identified in Santa Barbara, which seems to play a role in helping these get into the cells. But if you get rid of that receptor, they can still get in by another route. So there's at least two mechanisms. So, we know there's at least one known mechanism, but there might be others. I should say this is one of the ideas for current therapies for Alzheimer's disease, is to add antibodies which will actually bind to these seeds when they're outside of the neurons and to compete for them getting into the neurons to try to prevent them from propagating on and on and on. And there are clinical trials going on to do that. But
Is there any interest in blocking the receptor, or is it have a lot of?
yeah, so the receptor, what Ken showed in his paper a couple of years ago, or about a year ago, was that if you actually block that receptor by genetic tools, you could actually reduce tau propagation in some models. So I think, if you could understand all the receptors, or even just one of them, these diseases take decades to develop. So you may not need to stop everything complete. If I can slow this down twofold, instead of you getting the disease at 85, maybe you get it at 105, and that's a pretty good outcome, right? I'd take that. So yeah. Anybody but Larry. Yeah.
So Roy, the telomerase situation, when you block with that small molecule that had been developed for something else, why doesn't that cause problems?
Yeah, so I think it doesn't cause problems because our understanding of this pathway, that this pathway here where you add these A's, this is a signal to degrade the RNA, right? It says, this is a bad RNA, destroy it. And it turns out, you don't have that many bad RNAs usually. So there's a small number, right? And so if you stop degrading all those bad RNAs, it's not so bad, right? Maybe you have a bit of a cluttered cell, but it's not enough that causes problems. That's our interpretation. So.
Do you think that the story about woodpeckers as analogous to football players suggests treatments since woodpeckers don't get
CTE.
Alzheimer's?
Okay, so what Larry's asking is, what about woodpeckers? They're banging their head on trees hundreds of times a day. Why don't they get the same problem that football players get? And the other one to think about is bighorn sheep. They crash their heads together all the time. Why don't they get these problems? Well, it turns out that nature has solved this problem. If you look in their brains, the bighorn, they basically both have structures in the front of their brain, which, for lack of a better analogy, act as giant sponges. And so when the woodpecker's bill hits the tree and their brain goes forward, it doesn't go forward into your skull like ours does. It goes forward into this very spongy, heavily vascularized tissue. So nature has solved this problem. In fact, and the bighorn sheep do something very similar. So if we were to go into football players and implant a sponge in the front, and also you have to do the back too because you get a rebound effect when you hit your head, maybe we could solve this problem, but it's probably not worth it.
There are also amino acid substitutions in the prion-like proteins that prevent that from happening.
Yeah, so there are multiple different kinds of, in us it's tau which triggers this, right? And yeah, so.
Okay, last question here, and then got it.
Yeah, Shelley.
Hey Roy, great talk. When you get rid of that RNA binding protein, does it reduce tau formation? Tau aggregate formation, if you get rid of the RNA binding protein?
Megan, you want to answer that? No. So it turns out that RNA binding protein is a good marker, but it may not be the most interesting one. It's the one we discovered first. There are a couple proteins which we think have similar biochemical properties, which together influence tau aggregation in a positive way. If we get rid of the most abundant one, we see a small effect on tau aggregation, and we're trying to figure out if that's actually meaningful or not. And, stay tuned. We think the fundamental problem is we think that what the biochemical property of that structure is actually shared by multiple proteins. And so that's the tricky part.